মনে হচ্ছে আশিস এদিকে তাকিয়ে আছে।
ঠোঁট চেপে হাসিটা
একটু ল্যাগ হবে প্রনিবেশ দা, বিকজ
না ল্যাগের জন্য নয়, এতক্ষণ সত্যি আসছিল না ল্যাগটা, আমি জানি এবার আসছে।
Good evening, Suman. Chakraborty, that is. There are too many Sumans around here.
Good morning. Good morning. Good evening, Sovic.
Good evening. So, Suman's sound check হয়ে গেল. His sound is all right.
Okay.
দুটো স্ট্রিমিং অন হয়ে গেছে তো?
It's actually 8 o'clock. Should start.
হ্যাঁ, তুমি শুরু করো।
Somebody has to invite me. Should start.
স্যার কোথায়? সুমন দা?
হ্যাঁ, আমি ভাবছিলাম দীপ্তেন বাবু বা স্বর্ণেন স্যার এরা যদি কেউ বলেন আর কি।
সৌভিক দাকে ইন্ট্রোডিউস করুন।
হ্যাঁ। Okay, so thank you all for coming to this lecture series. So we are doing this in memory of Professor Ashish Banerjee, and in that regard, I would like to introduce Professor Soubhik Bhattacharya to speak few words about him.
Good evening. Thanks for joining. Good evening, everyone. It's a very interesting feeling, it's a mixed emotion. Ashish, of course, was my classmate for 5 years, along with many of my friends who are joining this evening. Let me try to, we can have the slide on Ashish, if you could. Professor Ashish Bandyopadhyay lecture series is something that has been conceived. It's a homage. It's a very sincere homage from the alumni of the Mechanical Engineering Department of Jadavpur University to commemorate his life and the impact of Ashish as a JU Mech, Jadavpur University Mechanical alumnus, as a mentor, as a teacher, as a friend, and of course as a colleague. Ashish has been a true mechanical engineer at heart, according to many, not just his classmates and his colleagues. He completed his bachelor's in mechanical engineering from Jadavpur University in 1981. Many of us did. And following stints at BHEL, Bharat Heavy Electricals, and Hindustan Motors and SAIL, Professor Bandyopadhyay joined Jadavpur University as a faculty member in 1987. He also completed a PhD at Jadavpur University during years. I happen to have a very sweet experience of being a co-supervisor of his doctoral thesis. Throughout his tenure at Jadavpur, he has been one of the most impactful teachers in the field of manufacturing, touching the lives of numerous students and colleagues. His untimely demise earlier this year in January left a huge void in the many lives where he had created an everlasting impact. The alumni of Jadavpur University Mechanical Engineering, we decided to remember him through a series of lectures across various domains of manufacturing science and industry, and also professional leadership and entrepreneurship and things like that in the coming months. Let me also, can you go to the next slide, please? Let me also share with you what is on the anvil, what we have. We can have the next slide. So on the anvil, we have 4 aspects, and this lecture series is the one we are starting today, both from academia and industry, the speakers. It will be on manufacturing, communications, pedagogy, and so on. And we're going to have at least one lecture in about a couple of months. The second one is to create, these are the aspirations, is to create an advisory group to help Jadavpur University mechanical students for future career steps. We're going to do some mentoring over there, bringing in real experts, practicing experts, help Jadavpur students with internship, intellectual support toward that, and also, as I said, a mentoring program toward research, toward business school programs, getting into that, and consulting career, and so on. We also would like to create a Friends of JU MEC task force. We want to form a team which will support JU MEC alumni, faculty, students, and staff across the board, all the stakeholders in unforcing economic hardships. We want to have a mechanism to pay back and create endowments, and also obviously to do the fundraising necessary thereof. And lastly, we also wish to archive Professor Ashish Bandyopadhyay's class notes, transcripted class notes, and have a good edit done, maybe to a professional publisher, and hence publish a book. So the next lecture topic we have in mind, we are announcing it today. It's on industry career paths for engineers, consulting. We probably will have it end May. Of course, stay tuned for the announcements. We will get back to you with a specific announcement on the schedule. A couple of speakers we have in mind, and they will be, as you can see on the screen, Prabuddha Guha, who is a JU MEC 2012 graduate. He's with the Boston Consulting Group and formerly with Accenture Strategy. I am Bangalore alumnus. Having experience in strategy and management consulting, and also in the energy area. The second speaker on the same day will have Shoumodip Banerjee, younger, recent graduate, 2015 Jadavpur MEC BME, and currently with Airtel, and also former Deloitte. He's an MBA from FMS Delhi, and is an expert in supply chain and manufacturing operations. Okay, that brings us to today's speaker, Dr. Suman Chakraborty. Without further ado, let me have this huge pleasure. I have this interesting situation here where both Suman and I, we graduated from the same department, Jadavpur MEC, that is, and then Suman has been a long-time dear colleague from Kharagpur, IIT Kharagpur Mechanical Engineering Department. So it's a tremendous honor for me, as I said, pleasure and fortune as well, to introduce Suman to you all, this august audience. It's also a joy for me, as I said, having been his colleague since he joined IIT Kharagpur as assistant professor quite a few years back, decades back. We have co-taught many courses and had the pleasure of serving in a large number of academic committees of IIT Kharagpur and beyond. I also fondly remember writing a brief bio sitting in a section committee meeting of INAE, Indian National Academy of Engineering, for his INAE fellowship application. So those are beautiful memories. And so let me proceed. Suman is a professor in the Department of Mechanical Engineering at IIT Kharagpur. He's a Sir J.C. Bose National Fellow, as bestowed by Department of Science and Technology, Government of India. He has been an Institute Chair Professor, currently Dean of Sponsored Research and Industrial Consultancy at IIT Kharagpur. Formerly, he was the head of the Department of the School of Medical Science and Technology, IIT Kharagpur. Suman had been an undergraduate student of Jadavpur University's Mechanical Engineering Department during 1992 to 96. Professor Ashish Bandyopadhyay was his teacher in one of the first courses on manufacturing science that exposed him to the world of manufacturing processes. Suman has been an extremely beloved student of Professor Bandyopadhyay and was in regular touch with him until the novel coronavirus outbreak. Suman did his PhD from IISc Bangalore in a record short duration. I don't want to get into the details. Talking about Suman, you need hours. Suman's current areas of research focuses on microfluidics, nanofluidics, and related innovations in diagnostic technologies for affordable healthcare care. And there is an echo happening. In addition to his proven proficiency in impactful scientific research disseminated in the form of publications in extremely elite journals, he has proven a unique expertise in technology development for the underserved population and community healthcare, including technologies transferred for commercialization. We will hear more about that, I believe, tonight. The frequency at which Suman receives top awards is actually quite awe-inspiring. Truly an academic and researcher par excellence. Let me share this very distinguished list of accolades with you. It's a very short list. I don't have time to go over his innumerable awards and recognitions. So he is a winner and recipient of the following: Shanti Sharu Bhatnagar Prize in 2013, elected fellow of the following: American Physical Society, Royal Society of Chemistry, American Society of Mechanical Engineers, all the Indian National Academies of Science and Engineering, and he's also a recipient of G.D. Bill Award for Scientific Research, Indo-US Research Fellowship, Scopus Young Scientist Award for high citation of his research in scientific journals, Young Scientist Engineer Awards from various National Academies of Science and Engineering, Alexander von Humboldt Fellowship as well. Throughout this professional career and growth, Suman has always been deeply attached to his alma mater. Yes, alma matters to Suman and many of us. This memorial lecture is a tribute to one of his teachers who he feels has enabled a gradual transformation of his vision from scholastic perspectives to technology, innovation, and dissemination for the underserved, for example, in affordable healthcare. I believe he will be sharing with us the narrative of at least one such successful endeavor where the developed technology has already been transferred toward a commercially launched product for people to use. It's all the more relevant today because we are sitting in a pandemic situation where the second wave is doing havoc throughout the country, and the discovery and the translational technology development that he has gone through is extremely valuable to the nation. It's relevant in these trying times, as I said. In undergraduate thermodynamics class, we talk about efficient isothermal gas compression, right? Suman possibly will go over the isothermal technique, isothermal control technique, as opposed to temperature cycling in the more conventional virus detection techniques. I believe that will be covered. Suman, thank you so much for accepting our invite to be the inaugural speaker in this lecture series in memory of our beloved Ashish Bandyopadhyay. And the stage is all yours. Over to you, Suman. Thank you so much.
Thank you very much, Shobhikda. It has been really an honor to be introduced by somebody like you, who, by your own, stature, is one of the top academicians that the country has now. Today, of course, the platform is very different, and we are going to all together pay our tribute to Professor Ashish Bandyopadhyay. And my viewpoint is that there is no better way of giving him a tribute than to talk about some work which was inspired primarily by his teaching that he made to our class. So I will try to share my slides. So I'm requesting Suman to please maybe unshare your thing so that and I can So I will now share my screen, and hopefully it will work, and then we will go ahead. So I just want to confirm that my screen is visible at the title slide, so that
visible.
Yeah,
okay.
Please go on. Yeah.
Okay. So the theme of my talk today is advancements in manufacturing for making innovative diagnostic devices, but not just any kind of innovative devices, the innovative devices which are imperative and important for affordable healthcare. As I mentioned, this is a tribute to Professor Ashish Bandyopadhyay, who first introduced me to the world of manufacturing processes. I can still remember that that was my second year of studies in Jadavpur University in the year, somewhat in the second half of 1993, when Professor Bandyopadhyay came to our class and started teaching about a course on manufacturing processes. Prior to that, all of us till higher secondary level were reasonably interested in at least physics and mathematics, some also in chemistry, maybe some also in biology. We all came to engineering and we got into the respective departments, not because we knew much about engineering, but we thought that it would make us a good career, and the joint entrance rank was conducive to that idea. This is the frank picture that we all had, and it was not clear that what is engineering, what is core engineering all about. Then came Professor Ashish Bandyopadhyay. I can still remember each and every class of him on manufacturing processes. It was a class of primary manufacturing processes mostly, so casting, welding, and metal forming. Now, this is something which, at that stage I could not realize. This is very much important for all students who, at their student life, do not realize what is the impact of a particular thing that is being taught. And I was of no exception. At that stage, I could not realize the impact of that in the coming days in terms of not exactly the contents that Professor Bandyopadhyay taught, but the spirit of the contents. Keeping that spirit in mind, I will talk about a scenario where a different paradigm of manufacturing can transform the situation of healthcare that countries like India and many other countries of similar socio-economic background they are facing. This is a report published in the Economic Times in mid-2013. The data may have changed, but the spirit is very much valid, that majority of the healthcare amenities and facilities is skewed in the urban scenario, whereas the rural India, though having majority of the population, is deprived of the basic amenities and facilities in healthcare, even the primary healthcare. As Indians, we always complain. We never talk about a solution. We always say that there are shortages of doctors, nurses, different barriers to access, etc. Often we blame politicians, often we blame others. But what we often do not recognize is the role of technology. The conventional technologies, including the manufacturing technology, which work in high-end sophisticated environment, may not work for the rural purpose. On the other hand, technology is something which in the present days is changing a doctor's perspective in many ways. Imagine the recent advancements in big data analytics like artificial intelligence, machine learning, virtual reality, augmented reality, healthcare trackers, wearables and sensors, medical tricoder, genome sequencing, drug development, and all these have revolutionized the clinician's perspective. But we also have to remember that undermine the role of manufacturing. Even the other aspects which are associated with manufacturing like biomimetics, microfluidics, nanoengineering, robotics, 3D printing, all together these have created a major disruption in the way in which medical treatment is being administered and planned. Being a researcher in microfluidics, our group primarily concentrates on point of care diagnostics with one drop of body fluid. I'm just trying to simplify it to an extent that I can understand all of us who are sitting in this forum today are by training mechanical engineers, but at the same time, I mean, currently we are all in different areas, and it is therefore more prudent to give an overview rather than being over technical. So imagine that a patient has to travel a long distance, go to a healthcare center, get a medical test done, and often it takes hours to get the result. Just today, I have heard that in a place like Delhi, the RT-PCR test reports for COVID-19 is getting 72 hours to be made available to the patient after the swab collection. So if you see, this is for the capital of the country, one has to imagine that what is the situation elsewhere. Imagine a different situation where instead of patient going to a hospital, a portable hospital, which is a medical device in the form of a diagnostic kit, is taken to the patient. Maybe one drop of body fluid. In this cartoon, it is blood, but it could be nasal swab, saliva, urine, whatever. It is collected. It is introduced into the device where certain tests are being conducted. Often the tests are related to certain reactions which give a change in color, and that color is decoded by using image analytics, mobile app interface, etc., to come up with a recommendation of the test result. Of course, the real device is much more complicated, but I tried to give you an overview. So these are all portable devices, functional with extremely limited resources, easy to handle. One can fabricate these in masses in principle, and that is where the manufacturing challenge comes. At the same time, we plan those to be inexpensive to the customer, and that is where there is another manufacturing challenge. It should be such that it doesn't require a lot of infrastructure to do the testing work. The test must be rapid, easy to monitor, and to some extent it could be automated depending on the cost structure. So this backbone of testing is something which has interested my research group over the years to develop certain technologies. I have given a list of some of our patents in this domain, not complete list, but representative of certain examples, starting from estimating, say, blood glucose, hemoglobin, creatinine, calcium level estimation, and of course, more complicated scenarios like testing the antibiotic resistance. Irrespective of the particular application, the motivation is very general. Rapid, low-cost diagnostic tests with high accuracy that may be performed by unskilled personnel outside controlled lab, and that can help serving the underserved community. Now, where does manufacturing come here? Manufacturing has been a backbone of the advancement in civilization, society, and technology. It has always been that, and it remains to be so. No matter how much we can talk about cyber physical systems, artificial intelligence, etc., if we do not have a physical system, we never have a cyber physical system. If we do not have a physical system that is manufactured, we never have a system where artificial intelligence can be applied. So manufacturing remains imperative. Now, a class of manufacturing which was of course not a textbook material when we people were college students, is a manufacturing of small devices. And it all emerged because of the first generation devices, small devices, which were primarily motivated by the electronics revolution, the electronics chips, the semiconductor industry. But it was all realized that these kinds of manufacturing processes, I will give a glimpse of these subsequently, may not be conducive for making cost-effective products that can be used by masses at low cost. And that gave rise to the second generation of these small devices, which are miniaturized devices, but cost-effective devices. And that cost effectiveness is brought in by very, very interesting innovations in manufacturing technology. So I will start with the manufacturing perspective with a simple example. In the right hand side, I have purposefully given an example of a casting process which was taught by Professor Ashish Bandyopadhyay when we were second year students. In the left part of the view graph, what we see is a sort of miniaturized version of that thing, where still you have a mold. In the mold, you put a material and allow the material to get solidified and deposited to form a structure. But all in the microscopic and nanoscopic scales. And that is what is microfabrication or nanofabrication all about. So it's essentially similar concept implemented differently. So I can say that it is like casting miniaturized. Let us take an example. This is known as photolithography. Those of you who are conversant with electronics industry will know that this is a backbone of making chips. Now, it is also a backbone of making microfluidic devices or miniaturized devices or MEMS, microelectromechanical systems, where basically you make a structure or a template with light. Why you use light is because the basic length characteristics you can play with using the wavelength of light. And wavelength of light, normally you will have of hundreds of nanometers. That means you can control features over that range. So what you essentially do, I am not going through the entire thing because I promised I will not be over technical. But what I want to just simply tell so that everybody can appreciate is that you have a light which is falling on a piece of a material. This is a mask that you can see. All of us who are mechanical engineering students will remember something like a master pattern in casting. So this is equivalent to that master pattern in casting. So you have a mask, and so the mask is patterned in a way that it will mask the exposure of light over material which we call as photoresist material. Some part of the photoresist material will be exposed to the ultraviolet light and some part will not be exposed. Which part will be exposed and which part will not be exposed is dictated by the design of the mask. Now once that photoresist is exposed by the light, it can, two things can happen. One is it can become more integrated, or its internal cross linkage may become stronger, or it can become weaker. So with further exposure, what can happen is that that layer of photoresist which was exposed to the ultraviolet light, either that can get dissolved or that can remain as integrated, and the remaining part gets dissolved. So in that way, you can get either a structure like this for a positive photoresist or a structure like this for a negative photoresist. So you can make a small groove-like structure, which can also be like a microchannel. Now, the photolithography requires a clean room. Clean room is a very special room where there is lot of control of dust, other particles, etc. And with extreme control and high-end lab condition, although it is possible to maintain such conditions for laboratory research, it is not so easy to scale it up as a large-scale manufacturing process. So far, therefore, in India, we do not have any such scalable manufacturing process that uses that kind of a technology. Now, over the years, when the miniaturization has advanced, we have entered into an era where life sciences and medical sciences and applications related to life sciences and medical sciences have interfaced with the miniaturization and microengineering, which is like a micro bioengineering or nano bioengineering. Now, for that purpose, a soft material instead of a hard material like silicon is used, which is very much biocompatible. That material is a polymeric material called as PDMS. So for that, the technology of manufacturing is known as soft lithography. So what you essentially do is that you prepare a master mold using the photolithography, but once for all. And then what you do is that you pour a material just like casting. This is as good as casting, which is taught in undergraduate second year mechanical engineering. So this material, this polymeric material is poured in, it solidifies, and in the way it gives a structure which is a soft polymer structure, and it can be connected with necessary tubes, etc., to seal and make a closed microfluidic channel for certain applications. Now, I talked about the second generation of miniaturized manufacturing, which essentially harnessed on all these, but tried to make devices which brings the manufacturing technology of these small devices out of the high-end lithography labs. That is mechanical micro manufacturing processes. I will talk about one specific example which relates to medical diagnostic applications, because that is the theme of my talk today. So broadly, this is a small disc on which we can do pathological tests. So we call it pathology on a spinning disc or lab on a CD. So this is something, if you are conversant with a diagnostic lab, you will see that there is something there called as a centrifuge, a huge rotating system where by centrifugal action there is a segregation of various components of, say, body fluid. For example, different cellular components of blood, because of density difference, they will get segregated by centrifugal action. Can we develop a mini centrifuge? And that is what is essentially the spinning disc with which we can make computer interfaces and have optical readout of the data. Now, keeping in mind the focus of today's forum, I will talk about the manufacturing of these spinning discs. So this manufacturing is something which is a process that does not require any clean room, and this essentially can be done anywhere. So I will give you an example, which is a 5-layer disc made of CNC machining. So this is essentially making 5 different layers with the plastic material in which there are holes and channels, and all those closely bonded together to make a single disc. As I do not want to be over technical, I compare this with a burger. So if you see a burger, so this is a 5-layer burger. So you see the 3 layers and the 2 layers of meat in it, and this is exactly in the manner in which this 5-layer structure is done. So you have a top layer 1, which includes the venting holes, and also the bottom layer, which you can see in this CAD layout. The 2 and 4 are essentially pressure sensitive adhesive layers, and 3 is the middle layer, which contains all the chambers where by centrifugal force, when this disc rotates, the fluid will move and do diagnostic tests. So I will try to run a movie to show you how this device is manufactured and how this is used for diagnostic test applications. So let us try to run this. Let me see whether it runs. I hope it will run, and it may take a bit of time. So this is it starts with the design. So you will see that you have different layers. I mean, all these are activities done in our lab. It's a reasonably small space in the lab, not a very big lab. And inside this we have small tables on which we mount these small machines. We do not have any special dust control in the lab. So what we are doing here is that we are making these 5 layers and bonding them together and putting an electrochemical sensor on the CD. So what this sensor is trying to do is to do certain blood tests as the disc is being rotated, and so it is giving a reading of impedance, which tells specifically that what is the level of certain analyte in the blood. So this is what is an example of this, which we call as blood on a spinning disc. So we can do different blood tests on this device with this manufacturing technology. And in the last year, we developed a technology around this disc for making a complete blood count test, or the full blood count test that is commonly recommended by doctors. Now, one side is the diagnostic test. I will of course talk more about that, but I would like to also talk about another aspect of this kind of research where manufacturing plays a very big role. Nowadays, there is a lot of advancement in making engineering devices mimicking what is there inside human body. The whole objective is to make a human body mimicking or human microvasculature mimicking structure in which you can make certain drug trials, maybe during the drug discovery process. If you can do it, there are lots of advantages. Normally, drug trials are done with preclinical and human trials, and these in totality span over almost like 15 years before the drug comes to even close to the approval stage. So these drugs are trialed on animals and then with a limited capacity on humans. Now all these have certain issues. Nowadays there are lots of ethical concerns in doing trials with animals, also doing trials with humans, and scientifically there is a lot of difference between the physiology of animals and physiological conditions in humans. Therefore, many drugs which may work in a certain way in animals may work differently in humans. On the other hand, if you make a complete engineering device without looking into the biological intricacy, then the lack of similarity between those devices and what happens inside human body may make the results not very much applicable when they are being used to predict what is the efficacy, for example, the drug as it is administered in the human body. So the first backbone of this is to manufacture deformable microchannels. So these are targeted to be deformable in a similar way as what we have microcapillaries or microvasculature in human bodies, that essentially is a network that connects the arteries and veins. It is not so easy to mimic that realism in biology, but one can make deformable microchannels, and in our lab we have standardized that to a good extent, and this is how we do. So again, this is like a casting type of manufacturing process. So you have a material called as gelatin, which is a derivative of collagen, a protein in human tissues. This is much more biocompatible than the PDMS that I have talked about, and it can closely mimic real blood vessel environment. And if you just line these with endothelial cells which are there in the blood vessels, this can actually very, very closely mimic the blood vessels in human body. These have very low elastic modulus and hence deformable. The method is very much similar to soft lithography and also very cheap. Just the PDMS is replaced by the collagen type of material, and using a simple needle lumbar puncture technique. What you can see here, this kind of cylindrical microchannel. So what is basically done is, when you have a needle and if you have a mold-like structure around the needle, so you create a cylindrical hole, and that cylindrical hole is what is mimicking the blood vessel, the microcapillary in human body. Now the real microcapillary structure in human body is much more complicated, because it is not just a single channel. You have a microvascular network, that is a network of channels. So how to make that? Now we have developed a manufacturing technique by drawing inspiration from the nature. So if you take the structure of leaves that is there in the nature, you will see different capillary branching structures, and there is a particular law in which the leaves in nature are made. So we tried to develop a hydrogel molding technique, again a casting inspired technique. I'm repeating on various derivatives of casting because I can still remember the classes of casting that Professor Bandyopadhyay taught to us. At that time, I didn't realize that these are going to be very imperative over the years. So in the process, we developed a microvascular like structure, and this is a very recent work. This work got published this year only, so it is a very recent work, and we could line endothelial cells inside this, and the way in which the cells behave in these channels, we could see exactly it is in the same manner in which these are reported to be in real blood vessels in human body. So where we are progressing in this, we are trying to sort of develop a biomimetic environment where we have certain organ representative structures, or you can say collection of cells, maybe heart, lung, kidney, liver, gut, like that, and connecting that with a network of micro channels, which as a schematic these green lines are shown. Now these will all be the biomimetic microchannels. So in that way, we are of course dreaming now, but we hope it will be a reality that we will be able to mimic exactly whatever is the microvasculature structure inside human body in a device, in an artificially engineered device on a chip. Now, that is of course some high-end science, high-end technology, which is progressing. It is progressing to an extent that we hope that it will create some impact over the years. However, society needs certain urgent interventions. The entire world is suffering because of lack of access to basic diagnostic tests. And these diagnostic tests are to be implemented at places which in reality are virtually green fields. So the point of care devices, which are fancy electronic devices which may work nicely at our homes for people who can afford, those devices may not work under these kinds of challenging conditions. So the paradigm shift is from point of care diagnostics to extreme point of care diagnostics. So extreme point of care diagnostics is a disruption in the way in which diagnostic tests can be done in extreme underserved or under-resourced locations. How can we do, or how can we manufacture these diagnostic kits? I will start with a simple motivation of using maybe a paper, pencil and eraser. These are sort of certain ingredients with which when we start studying in school, we never realize that these can be manufacturing tools and workpieces and can give us solutions that can impact real life. In the bottom right view graph, you can see there is a piece of paper which is soaked in glycerol, a glycerin type of fluid. Now, once you drop blood, one drop of blood on this paper, you see that the blood is spreading diffusively around it, and it is creating a very interesting pattern. In fluid mechanics, this is known as fingering instability. That is, a less viscous fluid, in this case blood, is displacing a more viscous fluid and creating certain patterns. While that pattern is interesting for giving certain recommendations regarding the pathology of blood, that recommendation will be primarily indirect. But to give a direct recommendation, what we have to do is to at least allow the blood to react with certain chemicals to have a reaction in the same way in which reactions are performed in pathology labs. So the blood cannot be allowed to spread just like that. Therefore, the blood, for example, if the blood is dispensed at a certain location, it needs to be constrained to go to a reaction pad where certain reagents are there, and the reagents will react with the blood to create a change in color. So to drive the blood from the source to the destination, we require a channel, and that channel is typically a microchannel, because then by capillary action, without any pumping force, purely by surface tension driven flow, the blood will move. So that motivates the manufacturing of paper microchannels or lab on a paper. Question is, what kind of paper? Is it a paper on which we write routinely? Maybe that one. But currently, we are using a more controlled version of the paper where the porous structure is more controlled. So normally a filter paper or a nitrocellulose paper, and the objective will be to make microchannels on the paper as per a design to allow the body fluid to move to a station where the pathological diagnostics will be done. So how do you fabricate these channels? Again, you see the photolithography, the wax printing, inkjet printing, and so on. So accordingly, although paper is a very, very simple and low cost platform, various manufacturing techniques have evolved over the years that can make microchannels on paper, like photolithography, laser treatment, inkjet etching, wax printing, flexographic printing, plasma treatment, laser treatment followed by plotting. I mean, so many other processes are there. The challenge with all these is that these require expensive instrumentations, specialized chemicals, and therefore may not be suitable in a low-cost paradigm. For example, see how the photolithography can be implemented on a paper. So you can prepare a mask, but the mask requirement, the mask manufacturing is not very costly because you can do it on a piece of transparent sheet. Exactly the transparency sheet. When we were students, before the LCD projector came into picture, this is the OHP, this overhead projector that was used, and these kinds of transparency sheets are very well known to all the students who were students at that point of time. So these are reasonably simpler than the photolithography done for the semiconductor industry, but it has a level of sophistication which cannot be implemented so easily in an uncontrolled environment. So from our lab, we disrupted this. We came up with a manufacturing process where we simply made a design like this. I mean, it could be more complex design in an office software, printed that on both sides of that special paper, and then introduce one more magic step, that is heating that paper on a hot plate at about 200 degrees centigrade for 4 to 5 minutes. The whole idea is that the toner particles can, which come out of the printer, they melt and block the pores in the black region. And fundamentally, in terms of science, it creates a hydrophobic barrier. And because of this hydrophobic barrier, the blood or any body fluid that will not spread around, but will be constrained to move along the white channel. In this method, no mask is required, and as such, no equipment is required. The method of hydrophobic barrier, we also tried to convert into a school level art by using a whitener ink pen or a correction ink pen. So the whitener ink has certain special particles which are hydrophobic. So if we simply make a hydrophobic barrier with a whitener ink, we can confine the fluid and do certain reactions there to come up with the diagnostic recommendation. The piece of paper that we talked about can be even more complex when it is not just a single type of porous material, but a collage of different porous materials all binded together to form a device called as lateral flow paper strip. This is exactly the kit that is sold in the market as a rapid antigen kit, which is used for many diagnostic tests. So it is a collection of various grades of paper and filters and membranes, and you drop the body fluid, which is say it could be blood, saliva, swab, etc. It would so whatever are the antigens, for example, it will go to the conjugate pad where you have nanoparticle conjugated antibody, and if the antigen is there of that particular virus, say, it will bind with the antibody, and the antigen antibody nanoparticle, this conjugate, it will move by a fluid flow which is triggered by surface tension or capillary force. It will go to the detection zone where that will bind with another secondary and primary antibodies, and that binding, successful binding, will make a line appear, one line to indicate just whether the fluid flow is occurring. So that is called as a control line. Another line just to indicate whether that particular antigen is there or not. So when both of these lines are occurring, by naked eye or by smartphone based examination, it is possible to tell whether the person is having a positive or infection or not. And the entire fluid flow is controlled by this absorbent pad, which is again a special grade of porous material. So all in all, you require to make different structures of different channels on pieces of paper, and deposit certain materials on the paper. I told you that, well, I mean, we can make, make it a sort of school art by printing on a paper, but that is not a scalable manufacturing. So how can you make a scalable manufacturing out of it? So this is a manufacturing plant which we have established at IIT Kharagpur itself, where we have certain machines which are conceptually doing the same thing what was shown in the previous slides as a lab scale technology. Now this is a technology upscaling for manufacturing, upscaling and commercial deployment. So you have a paper shearing, forming, assembly line, and you have a lyophilization unit, where what is done is on the piece of paper, basically you have a supply of paper. The paper is sheared and cut into pieces, and certain materials that are deposited in using this lyophilizer, and after they are dispensed and deposited, there is a vacuum package machine which makes it a complete package. So starting from the raw material of a paper, it becomes a complete diagnostic product in this entire semi-automated environment. Now, what do you want to deposit on this paper? Again, a simple lab research told us that you can make this paper electrically functioning, electrically sensitive if you deposit graphite. Graphite deposition is normally a costly affair, but what we simply did, I mean, this is a frugal manufacturing at its best, is just sketched with a pencil, maybe a 2B pencil on the piece of paper, and you can see the black region. This pencil sketch deposits the graphite which is there on the tip of the pencil, and it forms an electrode. So in this way, from our research group in the year 2012, we introduced a new branch of microfluidics called as paper and pencil microfluidics. The whole idea is not to use such pencil sketching for upscale manufacturing, but come up with a principle that can be upscaled, where instead of pencil sketching, you can make a deposition of graphite exactly in the same way in which the pilot manufacturing facility showed us the idea in the previous description. Now these microfluidic test kits need to be integrated with certain electronics, and this is where interdisciplinary engineering and manufacturing is so important. So the electronics integration has to be there, because all the readouts in the medical devices will have certain electronics backbone, which will essentially have some amplifiers, filters, maybe analog to digital converters, microcontrollers, etc. All embedded in a device architecture. So I'm just showing you a very simple version of this, which was made by our colleagues and students for a hemoglobin detection device. This is a simple electronic fabrication and assembly. What I intend to impress upon here is that mechanical manufacturing interfacing with this kind of electronics manufacturing and assembly together make a complete medical device, a prototype, which one such early prototype developed by our group was called as med colorimeter, which included this basic electronics, all embedded in a box, which was at that point of time made using the 3D printing technology. These prototypes have basically colorimetric detection principles. We could convert these into fluorescent detection with additional filters, but normally they will have a wavelength or an illuminator, typically say a red light maybe, or LED light with certain intensity, and it will have an optical detector, RF carrier or a protocol, and a digital resolution with certain bits or samples per second, and it can interface with a custom-made mobile app, and the voltage and the power requirements, they are very, very minimal. Now, how do you convert an image of a color reaction? That is, you have the blood or the saliva, whatever, it reacts with certain chemicals to change a color. Now, how do you actually interface that with the electronics to give the result of the test? So the color has to be analyzed by image analysis algorithms and has to be converted by some calibration into the biological information or the pathological information. For example, what particular color intensity corresponds to what amount of glucose or hemoglobin in the blood. So that requires image digitization, feature extraction, machine learning, etc., all embedded in the form of an image analytics app. And that is where, data science, machine learning, etc., can be interfaced. Now, on one side, we have a low-cost medical device or in the form of a paper strip. On another side, we have a electronic readout unit, but the challenge continues. Can we have something which even can replace the paper in the future years? And for that, a simple piece of cloth or a fabric material holds a lot of potential. The piece of cloth has a very similar cellulosic structure like a paper, but it doesn't disintegrate so easily like a paper. A paper, when it becomes wet, it tends to disintegrate. On the other hand, the piece of cloth doesn't disintegrate. The cloth pieces are also widely available. They have reasonably high tensile strength, flexibility, wonderful biocompatibility, and they can be used as armband or wristbands in a way that they can be made as wearable sensors. So we started with something which is very interesting, that again, the philosophy of making the microfluidic devices on this piece of cloth is very much similar to what we do on a piece of paper. That is, you make a hydrophobic barrier across which the blood or any other body fluid will not diffuse and spread randomly. So to make that barrier, we simply used a wood finish, which is commonly used by carpenters with linseed oil and mineral spirit. This is something which is commonly used by carpenters. And then with a simple paint brush, it was possible to make these structures almost manually and dry in open air for about 5 minutes at room temperature. So this is the manual process, but it is possible to upscale it either manually through a handicraft or in a semi-automated or automated manner by a mechanized system. So we try to make different structures out of it. You can see that there is a structure and you can see the blood is diffusing in the structure exactly the same way in which paper channels work. This is a cloth channel, and the hydrophobic barrier made of this material is working so nicely that no blood is actually diffusing. You can make very interesting structures. In fact, this would be a good school project for children who can paint different structures on these pieces of cloth. While blood testing and swab and all those things can be done on this, a very interesting diagnostic testing that can be done on these is using the sweat. Why the sweat-based testing is likely to emerge and grow is because blood testing requires penetration. It requires a piercing of the body to get, even if you want to get a drop of blood, it cannot be done without finger pricking. If you want to take a nasal swab, it has to be, it has to create a sensation inside the nose. Those of you who have done, who have seen people doing COVID tests in your family members, you will recognize that it is not a very comfortable situation. But sweat-based things, they do not require any collection of body fluids like this. So you can simply wear these as armbands or wristbands, and it can be used for non-invasive monitoring of certain electrolytes and analytes for infants and elderly people. This has a high potential of emerging in the form of a wearable sensor. And although there are many ways in which these sensors are currently being fabricated and used, these are actually quite sophisticated technologies, not something which can be used by for rural conditions in terms of manufacturing and use. Now, it holds the potential of having a different style of manufacturing via handicraft revolution. Nowadays, we say Make in India or Atmanirbhar Bharat. It is a very important mission according to the mission of our government, spearheaded by our Honorable Prime Minister. The entire tradition started with the era of Gandhiji, when it was an era of handicrafts. It may be possible to adapt that in the form of a modern, semi-automated or semi-mechanized and partly human interfaced, manufacturing paradigm to make such kinds of wearable sensors that can transform the rural economy and that can transform the way in which diagnostic tests, basic screening tests can be spread all around. While all these are different styles of manufacturing, very much different from what is taught in our undergraduate texts, the common objective is same what has been there in the traditional mechanical manufacturing, to bring these technologies out of academic institute labs, workshops, etc., to markets, to a manufacturing ecosystem where we have specialty materials, design and automation, frugal manufacturing, and digital convergence, all together with proper supply chains and engagement of micro, small and medium enterprises or different emerging manufacturing units with proper training, characterization and testing support to develop an indigenous ecosystem for new generation manufacturing of medical devices in India. What can we do with such simple devices is really wonderful. I will give certain case studies. This is an example where by using one drop of blood, we can do a large number of tests. This is a device developed by our testing team to work on various blood tests like glucose, hemoglobin, etc. One of the technical things here is that many of the blood tests require the separation of the plasma, the watery component of blood from the blood cells. And we have a very simple membrane-based technology where on a paper strip, combining microfluidics with the manufacturing and using the filter in a very judicious way, it is possible to separate the plasma from the whole blood. So you can see that the red colored thing is the whole blood with rich cells, and the plasma is what is diffusing and reacting with the reagents to create a change in color. The other example, I'm sure many of you have been waiting to hear about this, is the nucleic acid based testing at point of care. Currently, we are going through the so-called second wave of the novel coronavirus pandemic. The second wave has impacted India in a very bad way. There could be several waves of this kind, but what it all requires is adherence to certain basic regular life hygiene, maybe access to vaccination, and also access to testing and more testing. The nucleic acid based testing, which is the gold standard testing or the RT-PCR test, which is used to do these diagnostic processes in high-end labs, it has certain steps. One is the collection of swab from the patient, then extracting the RNA, which is itself a laboratory intensive process, and transferring the extracted RNA to a machine called as real-time PCR machine, where thermal cycling takes place. What the thermal cycling does is it amplifies one DNA to a large number of copies, what we do exactly in a photocopy machine. And the large number of copies of the DNA make it amenable to study whether the test genes which confirm the presence of the virus, whether they exist or not. So there is a complex thermal cycler and a complex optical system that makes the real-time PCR machine an extremely celebrated but costly and high-end machine that needs to be there in an infrastructure intensive environment for doing the RT-PCR test. In that way, it doesn't matter how low is the cost of the test kit is, the lack of availability of the resources, the technicians, the infrastructure makes it prohibitive to get the RT-PCR test done. And that is why, irrespective of the cost, people are now having a crisis of getting the test result. So many people are lined up with the test that a test, RT-PCR like test is taking 72 hours to get the result. At IIT Kharagpur, we tried to create a paradigm shift by substituting the celebrated real-time PCR machine with a simple COVI-RAP machine, which is entirely designed and developed at IIT Kharagpur. I will try to run a demonstrative video to show you how it works. It doesn't require a lab. What is done is simply the swab is collected. No separate RNA extraction is required in a separate unit. The entire activity by certain isothermal processes. This is again a fundamental thermodynamics and heat transfer process that goes on inside this machine. And at the end of that isothermal process, you come up with a smartphone enabled interface that tells you whether you have the novel coronavirus or any other nucleic acid test based infectious disease prediction, whether that disease is there or not. This is something which we feel can be a game changer technology in a way that the entire test could be conducted at any place where resources are limited. There is no need to have a specialized technician to do the test, and the results come out pretty quickly. So I will try to run a demonstration of this test so that you will understand what goes on. So what you see here is a demonstration in the lab. So this is a device which is there, a simple box around 300-400 gram. You require a tabletop. So the sample is collected from the nose. It could also be collected as a saliva sample, and then it is transferred into a tube where certain chemicals are there. Put in the machine. You can see all the steps and the subtitle. This is the isothermal temperature. The sample is brought out and it is mixed with the test kit material which is prepared by us. So this test kit material will be mixed. You can see that these are being mixed, and this is being put into the machine. So it will be put into the machine now, and that's all. There is no more manual step. A particular pre-programmed heating step will go on for about 30 minutes, and after that, once the test is over, what we do is we take an image, a smartphone picture of the products. These are the products. We don't have to take out of the tubes. We just have to take an image, and the result finally confirms in the mobile app whether it is COVID positive or the COVID negative. So all in all, it's a very simple test, and not just such simple tests for COVID purpose. We also developed other type of devices with very interesting manufacturing technologies, which because of interest of time I will not talk about, but I will just talk about an example where we have developed such a portable device for early screening of oral cancer. Oral cancer in India is a curse. It is it is mainly because of lifestyle factors in the underserved community, and normally it starts with pre-cancer stage, and it ends up with a more deadly condition. But if it is intervened and detected early, that could be managed. So again, I will try to run a very small video demonstration. So this is a simple plastic-based manufacturing product, which is interfaced with an electronics, but it could also be interfaced with a smartphone by replacing this electronics. So what it essentially does is we have a remote processor where thermal imaging inside the mouth, that is the temperature field inside the mouth. All of you are mechanical engineering graduates, will recognize that you can have, based on certain boundary conditions, certain temperature field. Here it's an inverse problem. It's a temperature field that is obtained by the imaging. The question is, what could be the blood flow in the tissues that could give rise to that temperature field, and that blood flow is correlated with the tissue conditions. So it's an inverse heat transfer problem that is essentially solved. So all in all, whether it is a blood test, whether it's a swab or a saliva test, or whether it is a test based on imaging, all developed with affordable manufacturing technologies, it is intended to impact the underserved by infusing deep science and innovative frugal manufacturing technology with public health innovation. It is intended towards rapid, low cost yet accurate methods for disease screening, user friendly and smartphone integrable systems so that user doesn't have to do too much, functional even in poor infrastructure without requiring trained personnel, and all these are possible if you have a manufacturing upscaling and MSME empowerment leading to not only the large-scale manufacturing of these devices, but also that leads to employment generation, startup incubation, and rejuvenation of the rural ecosystem. So I will, this is one of my, final slides. I will just show you, how these are really implemented. I am not talking about any hypothetical scenario. This is one of the field studies we made in a place known as Barra district, Birbhum, near Shiuri in West Bengal. Now, these are rural people who have never done any blood test in their entire life, and our diagnostic devices were being implemented by frontline rural healthcare workers along with my own masters and PhD students to for getting, in this particular case, the sugar and hemoglobin tests for different patients. So you can see that simple finger pricking is done. It is not a venous blood, the typical blood collection system. The finger pricking is done by a specialized needle to get 1 drop of blood. And this is because this is being done in a validation mode. We were comparing the results with other well-known, more costly devices with the devices, the technologies developed by IIT Kharagpur. So you can see this paper-based strip. It is inserted into the portable machine exactly in the same way in which a credit card is inserted into a credit card reader, and it's a simple smartphone interface. The mobile app takes over. It does all the analytics, and almost readily it gives outcome of the result in the form of a display in the mobile. So these results were made available to the rural patients who were consulting doctors in the camp. So many people who have never done diagnostic tests in their entire life were waiting, and we were so happy to see that it could impact the lives of many. So this is a continuing mission. It is by no means over. The job has just started. All these so far have been possible with kind support of our various parent bodies and collaborative partners. Of course, our own institute, IIT Kharagpur, the ICMR, the Ministry of Education, the Ministry of Science and Technology, and other foreign funding bodies like the Royal Academy of Engineering. I would like to finally acknowledge a long list of people without whom this work would not have ever happened. We have to realize at the end, these are all teamworks. By training, I am a mechanical engineer. I do not understand much of biology. I do not understand much of medicine, and I never intend to also become a biologist or a medical specialist. What we all do is to prepare a teamwork where we have medical doctors, experts from biology, medicine and electronics, with a long list of students, postdocs, with various specializations, again, from basic science, applied science and engineering, even medical doctors. I mean, one such medical doctor is my PhD student, and healthcare workers, and even the masters and the undergraduate students. Well, as I told, that the work has just begun. I intend to end with this quote: the woods are lovely, dark and deep, but we have promises to keep and miles to go before I sleep, as told by Robert Frost. Thank you so much for your kind attention to this talk, and perhaps we can have some discussions.
Thank you, Suman. There you go, ladies and gentlemen, Professor Suman Chakraborty. Magical work, and it continues to amaze people around us, around IIT Kharagpur, throughout the nation. Today, he covered the fundamentals, the earlier part which led to COVID app. I don't think Suman spent too much time on COVID app today. That could be another opportunity, another lecture. But that's turning heads in the country today. It has been transferred, and I understand, and a couple of companies, and it's going to benefit us and simple people across the nation. Suman, would you like to respond to a few questions? We have a few questions.
Absolutely, absolutely.
Let me present to you in no particular order. You mentioned about the acknowledgement in your last slide. Let me start from there. One of the questions is, I don't have information on who placed this question, so I'm sorry about that. What are the challenges as collaborators, like engineers, physicists, and doctors, may speak different scientific languages? What are those challenges, and how did you address those challenges?
I mean, it's a great question. because this is the biggest challenge. So I mean, when I started, this work as a mechanical engineer, of course, I mean, like all of us who are here, I mean, we are all mechanical engineers, and mostly maybe there could be one or two exceptions. So biology was never taught to us as a formal course, and many of us, even in class 11, 12, have not studied biology. So our entire biology or life science was up to class 10. Where it was descriptive to an extent that perhaps it did not create much of an impact and interest to most of us. Now, when we have to interface with medical doctor and biologists, it is one of the toughest things, because they speak in a language and because they are all trained to work in a particular stringent protocol. Whatever they have learned as a protocol throughout their life, it is very difficult to suggest something until and unless they are themselves very open-minded. So first of all, the biggest barrier is terminology. So when a biology specialist talks about very complicated terminology, that is very, very distracting to engineers like us. But fortunately, I got collaborators who are very patient. I mean, when I kept on asking them questions, at some point of time, they became sort of fragile, and they were compelled to respond in simple terms. For example, I can give you, I can talk you about the COVID story. So I started collaborating with Professor Arindam Mondol in our School of Biosciences, who by training is a virologist. So when we started working, prior to this, I have never worked with a virus. We have worked with other diagnostic technologies, but not virus. So I first learned from him that the kind of what is an RT-PCR test and what is done summarily there. Now, whenever I kept on asking questions to him, very probing questions, he would always come up with an answer. The answer, almost tells you certainly that this cannot be done outside the PCR machine, because that is how biologists are traditionally trained. Now, at the end, I tried to probe him continuously to an extent that he was in a position to explain me that what fundamentally takes place inside the PCR machine and whether that can be simplified to an extent. So once that is understood by me, now as a mechanical engineer, I have learned manufacturing, I have learned heat transfer, I have learned thermodynamics, I have learned fluid mechanics. And I also have friends who know electronics. So we can all combine this to an extent to translate that basic understanding in the form of something which may or may not work, but it can be explored. So the whole idea of doing this kind of interdisciplinary work is not that we have to be a jack of all trades. That means that we have to all know engineering, biology, electronics, so many things. What we have to understand is a mutual respect. That means as an engineer, I must have a respect to biologists, maybe what they learn, though I may not be knowing. I'm sure my biology collaborators, they will have equal mutual respect on us as engineers who may be knowing different things but are not trained in the way in which they know biology. There will be electronics people. We do not know much of electronics. Of course, nowadays everybody has to learn some basic working electronics, but not the formal electronics in that way in which the electronics and computer science people know. But we have to build up a language in which we can mutually communicate. That means a simple interface where our respective domains are represented as black boxes in simple terms to each other, so that they can create an interface without much detailing. And if that can be done, I think that is how a collaborative work can give rise to a technology development.
Excellent insight. Thank you, Suman. Suman, could you please stop presenting? Could you? I think you are still presenting the desktop or whatever.
Yes.
Thank you. All right, that just gives a better view. The
Yes.
next question, I'll switch to a somewhat more technical one. So how did you line the microchannels with endothelial cells? That's the question.
Lining microchannels with endothelial cells is a very standard technology. So I can share with all the protocols. In fact, if you see the ACS paper, which I cited, there is a detailed protocol of that. But summarily, the challenge here is not actually lining the endothelial cells in any microchannel. These are special microchannel materials made of hydrogel type of substances. So if you line them with endothelial cells and administer fluid flow beyond a critical sort of a flow rate, there can be a sort of disintegration. So this lining can be completely washed off. So there has to be a stringent binding protocol, and that binding protocol is more of a surface chemistry than anything else, and this is a little bit more special than the normal endothelial cell binding which is there in other microfluidic systems. But that detailed protocol is there, and I will be happy to share with the person who has asked this question.
Okay, the next one, it's a different, can individuals be delivered these microchannels for personal projects from facilities at IIT Kharagpur? Is there any mechanism for that kind of transfer?
Absolutely, yes. It can be done. There is no problem with this. We can, you can give microchannels. The whole idea is that it depends on what is the purpose of the microchannel. So if microchannels are not intended for these types of purposes, normally these microchannels are not the microchannels which are studied for academic or research purposes. The microchannels which are studied for academic and research purposes are mostly the ones which are made by these clean room based MEMS lab facilities. So we are not actually, so let me clarify here, we are not actually making use of those facilities for this kind of research, because here the whole idea is to make frugal devices which do not require clean room for manufacturing. So if the purpose of the microchannel is to study the design of these devices, we'll be happy to give that. If the purpose is to make microchannels out of a clean room based facility, I am not in charge of that particular facility in our institute, so I cannot give any sort of remark or commitment in this forum.
Sure. Yeah, of course. Somewhat related, maybe just give us a brief additional response. Is it possible to manufacture microfab sensors, microfabricated sensors in scale in West Bengal?
Yeah, absolutely yes. So we have actually set up a pilot plant for making these paper strip sensors. It is established within IIT Kharagpur as a part of a flagship program by the DSIR, Government of India, under their CRTDH on affordable healthcare. So it is a research and technology development hub on affordable healthcare. The objective is to have translation of technology from the academic or the research environment to the MSMEs, so that they can establish similar facilities. So if any micro or small or even larger industry is interested to just sort of replicate this thing, this entire machine that I showed is actually completely designed by us, and one company has in fact one MSME has manufactured this machine. It's a custom made one piece machine available in the entire world. It is not, and this is absolutely made in India, so it is not brought from anywhere else. So any company who is interested to replicate this machine in a larger scale to develop certain manufacturing facilities for making these diagnostic strips in West Bengal, absolutely welcome. We can assist with all the know-how and technology transfer.
Right. Suman, the next one is a little longish, just bear with me. I'll read it slowly. It's a technical one. On the inverse heat map problem, focusing on that, is a blood flow map or distribution being generated from the temperature profile of the oral cavity?
Yes, absolutely yes.
Okay.
So let me give an explanation to this. Maybe the remaining question will be related to that.
There's a part to it. Let me just hear the first part response briefly. I'll come back to the second part. Yeah.
Okay. So what we do is that in heat transfer, there is an equation known as bioheat equation. So the bioheat equation is essentially a standard heat transfer, just a governing differential equation for heat transfer with an additional heat transfer term, which is related to this blood perfusion. So it is a source term in the equation. So that blood perfusion term, normally if it is given as an input, the temperature field is the output. Here the temperature field is given as an input, so the blood perfusion comes as an output and it gives a map of the blood perfusion. Now in medical literature, there is an established mapping of blood perfusion with the histopathological condition. So now, of course, a standardization of this is required. So we try to standardize this with the biopsy data by doing actually clinical trials, not even pre-clinical trials, in the Guru Nanak Dental Hospital in the state West Bengal, where for about 30 patients, we took this image, did a multimodal image analysis and compared with the gold standard biopsy. And the agreement with the prediction was greater than 99%. So the whole idea is that without doing biopsy, can you give a prediction which even for screening purposes, no doctor will at the end spare you without biopsy, before coming to any recommendation in cancer, because that is what is the gold standard. But at least before coming to that stage, can you develop a screening where you have sufficient confidence in the result of the screening? And this device is purpose for that.
The next one, I'll take just two more questions, okay, in the interest of time. How to enhance the response of enzyme reactions in lateral flow paper strip? Can we use this chip-like platform for more than once in hemoglobin test machine?
Okay, first of all, let me clarify, the hemoglobin test that I showed is not a lateral flow test. It's called as MuPad or micro paper analytical device, where it's a simple colorimetric test. So that means that the blood will react with certain reagents and directly change the color. The lateral flow test is a part of our COVIRAP version 1, where we try to test the particular gene by doing a binding of the respective DNAs with the complementary DNAs on the paper strip by a process called as DNA hybridization. But the question remains very much valid, although it may not be relevant to the hemoglobin test. The question is very much valid, and it's a very, very important question, because if that reaction for an antigen test, it's an enzymatic reaction, but for others it could be a different type of reaction, but it's a binding reaction. But the binding reaction is read out either in the form of a color or a fluorescent readout. So the mapping of the readout with the reaction will tell you actually how you can detect. Now that readout is often poor because of certain non-specific binding. That means that because of certain spurious reactions, the actual binding, whatever signal it gives, there are certain spurious reactions which are not actually the test reactions. They are also giving certain signals, and those signals are captured by the detection and they are giving the false result of the test. So that is why actually in rapid antigen tests we get so many of these false results. So the way in which technology is progressing is the use of special nanobioconjugates or nanomaterials, which will make sure that it quenches out all the non-specific reactions and only emits the signals of the specific reactions. That is where the the future research is going on, and some of my students are also working in that direction. But it is actually a combination of nanotechnology and microfluidics, because those nanoparticles will be moved by the capillary driven flow. It will go to the test line and control line where the detection will be made. So, the interface of those conjugated nanomaterials with the surface tension driven flow. Actually, there is a very rich science with all these. Although the technology is frugal, but the backbone science is extremely complicated and mostly not yet understood. So this is, of course, a ongoing research and a very, very pertinent area of research.
Right. Suman, the concluding question of the evening, you have been a mentor for many people. I personally know and beyond that. It's in that direction. So the question is, what advice would you give on how to start a fruitful collaboration to an early career researcher?
Again, a very interesting question. My answer will be a little bit of something which is not expected. My answer is better not to go for collaboration at early stage of the career. Because, see, as I told at the very beginning, collaboration sometimes, gives you a temptation of becoming a jack of all trade and master of none. So, you tend to be superficial, and that, you can afford to be at a more mature stage where you have a complete grasp on your own core domain. No matter, whatever we are testing, virus or whatever, whatever is the engineering platform based on heat transfer, fluid mechanics, microfluidics, with whom I am interfacing, as a professor in the team, my word is the final word what we talk about microfluidics. It is a very rigid thing. Whatever I say is final, and I do not allow anybody to change that, no matter how great that expert be in electronics or biology or medicine. Now, to come to that, in my early stage of the career, of course, Shobhikda had been witnessing that over the years. I have primarily concentrated on, understanding deep of, fluid mechanics and heat transfer over small scales, micro and nano scales. And my entire, work in my early career has been to be more and more thorough in terms of the fundamentals, in terms of, fundamental characterization, experimental, theoretical, computational, etc. To learn ins and outs of these with extreme depth and without compromising any kind of rigor. And then at a later stage, I came to more and more collaborations. And that is what I will advise to all my younger friends, that collaboration is fine, but better, it is always nice to be a part of a collaborative team, but better not to be a leader. All of us like leaderships. All of us want to be leaders. And nowadays, nobody wants to be a follower. So, and from college level, we give leadership trainings. We never give followership trainings. Actually, followership is more important than leadership. So, be a part of an interdisciplinary group, but be a follower at the younger days. Allow somebody else more mature and more experienced to take up the leadership. If you take up the leadership from the very beginning, there is a danger that you become a jack of all trade and master of none.
Right, I couldn't agree with you more, Suman. Let me just repeat that in a slightly few different words. I have been saying this to many people in different forums, that to do something very well and contribute at the interfaces, which is the demand of the day, you've got to be rock solid at the core. You can't do it at the interface without being really, really strong at the core. So that's what you said. Let me before I start thanking you profusely, let me request all the core team members to switch on their cameras for a few seconds, and let me. Can you all switch on your cameras, please? Suman, Dipten, Shorodip, and Pranivesh, and Sharnindu. And So, Suman, thank you so much. I don't know how to, nothing will be enough. I feel very good along with all these team members. I would like to, along with Professor Suman Chakraborty, I'd like to thank Sharnindu Sen and Dipten Mishra and Arkadeep Kumar and Pranivesh Mandal and Suman Sinha Roy and Shorodip Poddar and many others who contributed. And we do have a dream. We do have a beautiful dream, a narrative that we mentioned at the beginning of this evening, that we want to do something for this department, and Ashish should have given a big hug, a very tight hug to Suman today if he had heard this lecture. This is what I expect Ashish to do. Okay, he is not here, but we can do wonderful, creative, constructive, helpful, supportive things. That will be the best homage to a person like Ashish. I keep saying his first name because, I used to go back home or sometimes go to Jadavpur for 5 years in the same train, in the same compartment, sitting next to him possibly. So, those memories are rushing back to me, and it's just a sad premature loss, but let us convert this to something constructive. Suman, you become part of the core team. You have been. And so along with this lecture, you have contributed tremendously, but we wish to have you also contributing in the other various plans that we have chalked out. Now, let me hand it over to someone like Suman Sinha Roy or whoever else to have a closing word. Again, from my personal behalf and on behalf of the entire JU-MEC alumni, the entire family, Professor Suman Chakraborty, I thank you very much from the bottom of my heart. Over to Suman Sinha Roy, the other Suman.
Thank you, Shovikda. Thank you, Sumanda. And it's a great pleasure hearing your talk. And again, as we have always mentioned, Ashish Babu, when we called Asbi, some people were Ashish, some people were Ashishda. They was very close to all of our heart. And we are trying to forge a path to making a better, like a friendship circle among all the JU-MEC alumni. And I would really appreciate, on behalf of all the core committee, if you people who are in the audience, if you guys can come up, and if you want to take more responsibility on doing more things, please let us know. You have our email ID, which is asbmemorial@gmail.com. Please send us your suggestion and how you would like to contribute. Please let us know, and we'll be very, very happy and eager to work with you all. And I will hand over to any other from the core committee if any one of you would like to talk.
Dipten, we can't hear you.
Hi. Let me open this thing. I'd like to say a few words that we people from the academic domain, we bear a lifelong stigma from people at large that the type of work we do, they are barely having any connection with the societal need, the industrial need, and all those things. Now, Professor Suman Chakraborty, he has shown what being in academics people can do for society at large, particularly the underprivileged people. And so it will be a kind of a what you call leading example for people who will be at the beginning of their career to follow a path that has been traced by Professor Chakraborty. So that is for that we'd like to be very thankful to Professor Chakraborty. So,
anyone else? Okay, it's pretty late. Anyone else? Sharnindu, Arkadeep, Pranivesh?
I think everybody of us is mesmerized. So only to thank Suman. And it's really a very, very good start for our team, I think. And we need to keep it up for the coming days. That's all.
Before we all kind of conclude, I would just like to remind one more time is like we have another lecture coming up, which will be at the end of the May, which will be on looking at life for engineer in the consulting world, and we will have two speakers. We are trying to get another speaker, and both of them are Geomic alumni. And please stay tuned. We will send you an email with all the description. And at the bottom of the YouTube link from which you are sharing, you will see there is the Google form. It will be active. So if you have not filled it up, please fill it up. If you want to send it to your friends, please send it to your friends, because we would like to keep this community going. Just to give you an idea, this first lecture actually got us 378 people. We made an estimate. We are almost 5,000 people active right now from Geomic family. So we will try to reach each and every one of them. So your help and your contribution will be deeply and greatly appreciated. So again, we want to create this Geomic friend circle. We want to make our interlink really, really strong. Thank you.
So on that note, we'll close this evening. Thanks everyone, and it has been a personal pleasure for me to speak and introduce Suman. Wish we could do it in person, but we'll do it. Someday we will do it. We will overcome. Thank you so much.
Thank you. Thank you. Thank you, everyone.
We are closing it. Bye for now.
Bye. আচ্ছা, ইউটিউব লিংক গুলো বন্ধ করা হয়েছে?